So in today's lecture, we'll be discussing genomes and chromosomes. Now, it turns out, throughout this whole class, basically, we've been drawing chromosomes as sort of squiggly sort of lines or little strings of spaghetti or little snakes or what have you. But it's important to know that most of the time, that's not what chromosomes look like. So in this particular series of images, there's a particular DNA stain known as DAPI stains. DAPI stains DNA. Most of the time, cells would look like this, and so here's the nucleus, and so you can see that most of the time the DNA is diffuse. So in fact, it's all just sort of like squiggly threads, all diffuse within the nucleus, and it's really only during cell division, when the chromosomes condense, you have this sort of condensation pattern that we have, that we actually sort of see them as we've been showing them throughout this entire class. Now, what we're going to do now is we're going to kind of dig down deep into sort of, once you have the DNA, what is how the DNA is structured within the nucleus. And so the core structure of DNA is in a particular thread called chromatin. All right, chromatin is a combination of proteins and DNA. So remember that at the beginning of the semester, we talked about how chromosomes were known to have genes on them, but it wasn't clear. It was known that DNA was on chromosomes, but also proteins were on chromosomes. So it wasn't clear if it was the DNA of the chromosomes or the proteins on the chromosomes that comprised the genes. Now, obviously, now we know that it is the DNA sequences that comprise the genes, but there are many, many other important proteins that contribute to the function of chromosomes that we're going to discuss. The core unit of a chromosome is something called the nucleosome. And the nucleosome is basically DNA wound around important proteins called histones. All right? And histones come as octamers. And by octamers, I mean there's 1, 2, 3, 4, 5, 6, 7, 8. And actually, there's sort of two sets of 4 here. And so the histone names are histone 2A, and there's 2 copies of those. And there's histone 2B, and there's 2 copies of those. Histone 3, there's 2 copies of those, and histone 4. And they wrap around, just a little under 2 exact times. And between one nucleosome and another nucleosome are what is called linker DNA. And this forms basically sort of what is called the beads on a string model. Now, once we go from DNA, we have DNA wrapped around histones to form nucleosomes, and then the nucleosomes themselves compact with each other and form something called the chromatin fiber. Here's a drawing of the chromatin fiber. Here's another structure drawing of the chromatin fiber. And this particular configuration is usually about 30 nanometers in width, and so this is known as the 30 nanometer fiber. Now, at a higher scale, the 30 nanometer fiber forms looping structures, which we're not going to get too much into, but then will form larger chromatin domains. And this is a computer model for how we believe the chromosomes are structured within the nucleus during that stage of diffuse blue, what I showed in the first slide in today's lecture. And what we have here is basically the colors indicate the different chromosomes. And so there are different chromosome domains. And so, for example, whatever chromosome is comprising the red chromosome here has sort of an aggregation here, but there's pieces of that chromosome over here, and there's pieces of that chromosome over here. So what this means is that chromosomes tend to reside in territories within the nucleus. And one thing that's a kind of an interesting thing to note is that the interior regions we can't see, like deep inside this ball here, tend to be more active. And by more active, I mean more of the RNAs are being generated through transcription. And out on the periphery of the nucleus, gene expression tends to be reduced. So active genes tend to be localized inside the center of the nucleus, and then less active genes will be outside. And so the three-dimensional structure of the nucleus plays a role in determining patterns of gene regulation. Now, there's a way that we can actually begin to study the three-dimensional structure of the nucleus, and in fact, we actually do it through next-gen sequencing. So we've talked a little bit about next-gen sequencing that can be used to identify the genetic basis of rare diseases. For example, you can have a baby that shows up in the NICU and has a disease and you don't understand what's going on. You can sequence the baby's genome through next-gen sequencing, and you can actually potentially identify mutations. But another thing that people have figured out how to do is they can actually infer the three-dimensional structure of a nucleus by also doing next-gen sequencing. And you might be saying to yourself, like, how the heck do they do that? Well, the way they do that is you take a nucleus, here's a cell nucleus, and the first thing you do is do something called cross-linking. And cross-linking basically fuses the three-dimensional structure in place. It actually uses formaldehyde sometimes for this. And so once you freeze that three-dimensional structure in place, proteins that are nearby each other can stick to each other. And so here's chromatin, which is protein on DNA, and then we have strands of DNA. And what this stretch here is, and this stretch here is DNA, maybe from entirely different chromosomes or maybe from the same chromosome, that is in close proximity to the other strand of DNA in three-dimensional space. All right. So once this sort of like frozen image of DNA is obtained, by image I mean like actually frozen in a test tube, the DNA can be fragmented, so pieces can be cut here and cut here. There's a name of an enzyme called the restriction enzyme, which we'll learn more about later in the class. And if you cut the DNA in that fashion, and then what you can do is you can do this funny thing where you can actually fill the ends in, and you can use DNA ligase, and you can generate circular DNA molecules this way. Now this represents a circular DNA molecule representing the fact that these two DNA molecules were nearby in this three-dimensional nucleus. Then, if you fragment that and you actually are able to enrich for those junctions, if you sequence from this end and you sequence from that end, you'll identify reads using something called paired end sequencing, which you have one end and then another end. You'll identify reads that correspond to different parts of, say, for example, a chromosome or different parts of the genome that were in three-dimensional space nearby each other. And so this particular sequence might be like, ATG blah, 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 whatever. And this particular sequence might be GC blah, 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 whatever. And you know that this comes from this particular chromosome region. And you know this blue one comes from this particular chromosome region. And a high abundance of those reads that indicate a high abundance of reads from one chromosome region to another chromosome region in this, where you're sequencing millions and millions of reads, indicates physical proximity. And so you end up with an interesting plot-like chart like this. This is along the length of a chromosome here, and red indicates lots of reads in which one chunk is co-occurring with another chunk. And so, of course, along the diagonal, this indicates just the entire length of the chromosome. Well, this is actually a zoom in, but you'll get a read along the diagonal because you will have, pieces on one part of the chromosome will end up sequencing nearby other parts of the same chromosome if they're right nearby each other because they're physically linked on the chromosome. But you find these interesting things where this particular dot or this particular dot is on the sort of off diagonal here. And what that means is that this particular position, we have position 1, 2, 3, 4, 5, blah, 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 on the chromosome, same position, 1, 2, 3, 4, 5, blah, 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 on the chromosome. Let's call this position 5, all right? What this means is that, this would be position 1, 2, 3, 4, 5, right? And this would be position 1, 2, 3, 4, 5, 6, 7, 8, 9. So position 5 here, all right, is showing a lot of reads associated with, say, position 10 here, right? And the position 5 and 10 are arbitrary in this, but when you get these little dots on the off diagonal, you get these little bars here like this. What this indicates are contact points along different chromosomal regions, which are known as topically associated domains. So here are these chromosome territories which show up with the different colors, but if you were to look inside of this, you'll have these like looping out structures, which I mentioned, right? So this would be a looped structure of 30 nanometer fiber, and you might, for example, find reads here and reads here along a chromosome arm associating with each other, and that would be shown with these sort of dots on this off diagonal here. So this would be a region that tends to show up in the sequencer associating with this, and that is what is called an insulated neighborhood, and there's gene regulatory mechanisms that require this looping. So it's a next-gen sequencing approach that you can use to infer the higher order structure of the three-dimensional nucleus. So if we zoom out even further and we talk about just the chromosomes large-scale structure are basically broken up into a few different parts. Okay, you have the telomeres, which are the ends, right, and the centromeres, which aren't always in the center, but the centromeres are defined by the place where the spindle attaches to the chromosome arm. And then there are two kinds of types of chromatin, all right, the chromatin being the mixture of DNA and protein that is on that is making up a chromosome structure. So the one type of chromatin is called euchromatin, so U means true chromatin, and heterochromatin means hetero meaning different, so this is like a different type of chromatin. And these were observed, these types of chromatin were observed by cytologists, in the early part of the last century, who were using DNA stains to identify different patterns on chromosomes. Okay, so euchromatin, or true chromatin, has several properties which are important. One is it's gene rich. So most of the genes are in euchromatin. It usually has a high rate of recombination. During S phase, during DNA replication, it's the first part of the chromosome to replicate early. It's where the DNA replication forks open up. And it's got this general property of being, quote, open. Open meaning not very compacted, and open then therefore also meaning basically perhaps a higher activity of gene expression because there's a lot of transcription happening on the genes that make euchromatin gene rich. Now, heterochromatin basically has the opposite property. So it tends to be gene poor, it has a low rate of recombination, it usually replicates later in S phase, and then it's condensed as opposed to open. Now, we'll focus our studies mostly on human chromosomes. This is a karyotype made of fluorescent probes, and so each of the chromosomes from a human have been independently labeled with a fluorescent probe. So you'll have chromosomes coming in pairs, and for example, this chromosome, if my eyes serve me correctly, is the homologue of this chromosome, and this chromosome here is the homologue of this chromosome. And I'm doing that based on the color. So a karyotype oftentimes is more seen like this. This is the type of karyotype that all of us would have, provided to our doctors right the moment that we were born. This is a result of a banding pattern that comes from a particular chromosome stain. Your blood is taken at some point when you're born, soon after you're born, and mitotically dividing cells are identified, and they are stained and they are squashed on a microscope slide. And obviously the chromosomes don't splay out like this, they splay out in a chaotic fashion, and obviously most of the cells that aren't dividing wouldn't show something like this. It's the dividing cells that would show something like this, and then a person would take a picture of it through a microscope after staining the dividing cells, and then they would use basically Photoshop to identify, based on the banding pattern, the particular pairs of chromosomes. So in this particular case, they're all lined up. Before Photoshop, it was done with an X-Acto knife. So you have chromosome 1 in a pair, and chromosome 2 is a pair, so on and so forth. And the chromosomes are identified by size. Obviously, the larger chromosomes have numbers like 1, 2, and 3, and the smaller ones have chromosome numbers like 21 and 22. The size and the structure and then the banding pattern based on the DNA staining. So it's worth thinking about, this always kind of blows my mind. When do you think it was actually figured out that we have 23 pairs of chromosomes? Like, 20 years ago, 30 years ago, 50 years ago? How many? We have 23 pairs because, if you don't have an X combination, you have an XY combination, obviously. Well, it was only in the 50s that was actually figured out, so about 7 years ago. So even in the 30s and 40s, we didn't know exactly how many chromosomes humans had. Now, when we talk about these karyotypes, we can talk about the sort of structures of the chromosomes that we see. We give them names based on the position of the centromere. Again, we define the centromere as where the fibers attach during cell division. Here we have what is called the metacentric chromosome, all right, because the centromere is basically right in the middle, and a submetacentric chromosome where it's a little bit off, an acrocentric where it's almost at the very end, and then a telocentric where the telomere is also the centromere. And so you'll see these different types of chromosomes will show different types of patterns during mitosis and in anaphase. So in this particular case, a metacentric chromosome, because the centromere is right in the middle, will show sort of this V-shaped pattern. But a submetacentric will be more J-shaped, and an acrocentric, where the centromere is almost at the very end, will show more of like an I-shaped pattern in the anaphase. Now, we'll do a little bit of a tour of the 23 human chromosomes. Cytologically, the chromosomes are given the names. The arms are known as P and Q arms. All right, so most of the chromosomes have one longer arm and then one smaller arm, and the smaller arm is the P arm. And P, in this particular, the reason it's called P is for petite, and Q is not P. All right, so there is also those banding patterns that I discussed, and those give basically a sort of cytological definition of position on the chromosome arm. So this would be P1 region, all right, and then inside there are going to be bands that have designations like 13 or 21 or so on and so forth. All right. So you have large chromosome, chromosome 1, small chromosomes 21 and 22, and then the X chromosome and the Y chromosome. So let's talk about some interesting aspects of the 23 human chromosomes. So one very curious chromosome is chromosome 2, all right? And the curious thing about chromosome 2 is if you take, and then chromosome Y is also a little bit strange, and we'll talk about the X chromosome as well. So let's talk about chromosome 2, all right? If you take a fluorescent probe from chromosome 2, and you use it to light up a chromosomal spread from chimps or orangutans, you'll see an interesting pattern. So on the right-hand side, we have this cute little baby. Some cells were taken from the blood. The chromosomes were stained, and you can see here's chromosome 2, one of them, and then the other one's from mom and one from dad. But what's interesting about this is that you actually don't see this sort of pair pattern. If you look at this chromosome 2 from chimps or orangutans, you'll actually see, here's chromosome 2 with this interesting red tip here, and then there's another chromosome 2. So what the heck is going on here? Do chimps and orangutans have 2 chromosome 2s? All right. It's interesting to think about. So, there's 2 hypotheses, right? You could propose that maybe at some point in the ancestor of this little orangutan, our chromosome 2, or the ancestor of our chromosome 2, broke into 2 pieces, right? And so you'd have these fragments. The other possibility is that there was a fusion event, right? And so that these 2 chromosomes and these 2 chromosomes came together and fused in this fashion, and going from 4 as the diploid state to 2 as the diploid state for just that particular chromosome. So how would you figure that out? How would you figure out if it were a fusion event or a breakage event? Okay, well, the best way to answer that is to basically do this analysis in a bunch of other great apes. So you can do this in chimps and orangutans and gorillas and what are the other great primates? Baboons and or was it gibbons? Well, anyway, and if you look, all of those will have this particular pattern. All right, and so actually humans are the only ones that have the pattern in which they're together, and so that's more consistent with the hypothesis that a fusion event occurred. If everybody had this, if only orangutans had this pattern, then you might argue that it broke in their lineage, but everybody seems to have this pattern among the great apes, except for us, so that indicates a fusion event. Okay, and so chromosome 2 arose from a telomeric fusion of two short arms. And so here you have at the DNA sequence. These would be the telomeres. They would come together, and then eventually one of the telomeres has to, basically adopt the function of the centromere, and then the other one has to lose its centromere function, because if you have two centromeres, chromosome will get torn asunder. And so eventually one of them inactivated and you ended up with a short arm and a long arm, and that's where our chromosome 2 came from. The Y chromosome, as I said, is another unique chromosome. It is very famous for having very low gene density, being heterochromatic, and actually containing a very important locus called the SRY, the sex-determining locus. And it's the Y chromosome that carries the sex-determining locus that determines the sex. In the absence of, if you have a Y chromosome that's deleted for the SRY, it won't induce maleness in an embryo. It's thought that the Y chromosome evolved through degeneration of the X due to lack of recombination. So there is some recombination in males between the X and Y, and this is in this region called the pseudo-autosomal region. But aside from that region at the very tip, the rest of the X and the Y's don't recombine. Now there's a couple interesting things to think about. First off, why is there maintained recombination in the pseudo-autosomal region? And remember, in one of the earlier lectures I said that recombination is very, very important for ensuring segregation happens properly in meiosis. In the absence of recombination, non-disjunction can happen, and that can actually cause trisomy 21, right? A lack of recombination. We mentioned that in one of the earlier lectures. And it's interesting to think about, like, why. Okay, so if you have some recombination here, what's the relationship between the lack of recombination and the fact that the Y chromosome has low gene density and is heterochromatic? Keep in mind that in females, when they're females with their two X's, those X's are always recombining. So the Y chromosome is basically a chromosome that basically never recombines against the length of it, except for this little tip part. And it's, evolutionary genetic theory, and I won't get too into it. It's worth, you could go and read about it on Wikipedia or whatever. But when recombination is prevented on chromosomes, that leads to their degradation because they're not allowed to accumulate mutations that might prevent them from degrading. So what happens is the X and the Y are basically have a common ancestor. The Y chromosome, the sex-determining locus arose on the Y chromosome. Recombination was shut down to prevent genes from the Y chromosome moving onto the X, and then once that recombination shutdown occurred, then the Y chromosome began to degrade. Okay, that's all I'm going to say about the Y chromosome. All right, we'll spend a fair bit of time talking about the X chromosome. The X chromosome is an interesting chromosome because it occurs in a different copy number between males and females. It turns out that chromosomal dosages must be balanced, okay? So here we have a bunch of autosomes. So, 2 autosomes, 2 autosomes, 2 autosomes, 2 autosomes. This is, for the most part, going to be okay, right? Maybe this individual has a mutation, but aside from that, all of the chromosomes are in the proper dose. However, when there's a case where there's 1, 2, 3 of one chromosome, all right, and then 2 of the others. This can have very harmful effects, and the reason that it has harmful effects is because there are genes that are regulating other genes, which are regulating other genes. And when the genes that are regulating other genes that are regulating other genes, when they're in a different copy number, then they'll cause misregulation of gene regulatory networks that will cascade throughout entire, basically, modes of gene expression control. All right. So it's interesting to think about like why that would be, but that's the general principle. Okay, so we've decided now for autosomes having maybe an extra dose of one chromosome, or maybe, for example, a missing of the other chromosome, that can be harmful for an organism. Then what's going on here? What about this? Because in this particular case, males have one X chromosome and females have 2 X chromosomes. So why is the lack of an X not harmful in males? Why is it that, oh, there's an X missing, the Y chromosome doesn't have very many genes on it, the X chromosome is missing, why aren't the genes that are regulated by the X or even present on the S misregulated and therefore causing defects in males? And the reason that's the case is because the X chromosomes are differentially regulated than the autosomes. If you look within cells, right, you often see a patch of chromatin in the nucleus which looks all compacted. And this got the name bar body because this bar body, this compacted DNA patch, can be found inside the nucleus. And it wasn't known what that was when it was first described, but then Mary Lyon made the realization that that is an X chromosome, and the function of the bar body is to properly maintain the dose difference between males and females. So let's talk about what's going on. All right. So it turns out the X chromosome in females, one of them is turned off. All right. Now, so what that means is that males have an X chromosome and a Y chromosome, so they have one dose of the X. But in mammalian females, there's 2 X chromosomes, but one of them is turned off. Okay, and what that does is, even though now that means that gene expression in females is derived from only one X chromosome, it solves this imbalance between the sexes, in that both males and females basically rely on the expression of a single X chromosome so that the other genes in the genome can be regulated properly in both males and females. Now, how is one X chromosome turned off? All right. In human females, the way that it occurs is through a random type of counting event. So when a zygote is formed, here you have an X chromosome, one of them from mom, shown in red here, one of them from dad, shown in blue here. And early in life, all right, both X chromosomes are active. So this cell here, the zygote, is going to divide 2 times, and there's 1, 2, 3, 4 cells. But soon thereafter, what happens is X chromosomes become inactivated. All right, and so the long chromosome here that's all stretched out, that will be the active X, and the sort of squiggled up one will be the inactive X. And what happens here is that this particular chromosome, this X chromosome, has been inactivated. But in this particular cell, the other X chromosome, the X chromosome from dad, has been inactivated. And so here we have a scheme where we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different cells. And in each of these cells, it's either one or the other X chromosome that's been inactivated. The red one or the blue one or the red one or the blue one, so on and so forth. Now, it's a random event, and there's a lot of interesting research being done about how this random events are occurring and how one X is counted and the other one is maintained to be on. But what then happens is once that X chromosome is inactivated, say for example in this cell, the blue one, all daughter cells, for the most part, from that particular cell will have the blue chromosome inactivated. And over here, this particular cell, all daughter or descendant cells of this particular cell, the red cell will be inactivated. And this leads to something called the single active X principle, which is that all but one X is silenced. And this random inactivation is something that is interesting because it explains the observation of calico cats. So calico cats are only female. All right. Well, there's some weird exceptions where males might have an extra X chromosome for reasons which I won't go into right at this moment. But for the most part, all calico cats are female. All right. And the reason that they have a calico pattern is that these black patches and these red patches, those are patches of gene expression that cause pigmentation. In this particular case, one of the X chromosomes expresses black pigment, right? And the other X chromosome expresses this sort of reddish pigment. And so in this particular patch, all right, the X chromosome that expresses red pigment is turned off, and they just form this thing called mosaicism, where there's a patch of cells that would maybe be descendant from one particular X inactivation event. But then you have another patch of cells which are dependent on the opposite inactivation event. In this particular patch of cells, you have cells that the X chromosome that expresses the black pigment is turned off. And what that means is that you have basically a cat then that's a patchwork of different X chromosomes, in which case one is inactivated or the other is inactivated. Now, this problem of dosage compensation is seen across basically all of animals. Mammals have an XY system, all right? It turns out Drosophila also has an XY system, but the Y chromosome in Drosophila or other flies isn't what makes it male. What makes Drosophila male is how many X chromosomes they have. So if they have 2 X chromosomes, they're female. If they have 1 X chromosome, they're male, and of course there'll be a Y. But in both cases, there has to be sort of an equivalent expression of the X between males and females. Now, in mammalian systems, one of the X's is randomly inactivated in females, but in males, Drosophila, the way that the dose of the X is achieved in males and females is that the X chromosome in males is upregulated twofold. And so males basically have twofold expression from the single X, and that makes the expression level from both X's similar between females and males. It's interesting in marsupials, it's not random X inactivation. In fact, the X chromosome that's inherited from fathers is inactivated. So, females have 2 X chromosomes like, non-marsupials, but 1 X is always inactivated, the one that was inherited from dad. Nematodes have an interesting pattern of sex determination, well, specifically C. elegans. They are XX or XO, so these males don't even have a Y chromosome at all. O stands for nothing, it's just a single X. And so again, there's this dosage challenge, but the way that the dosage challenge is met is that in females, there's a 50% reduction of the X, and so it basically gets dialed down, and that ensures similar expression of the X for males and females.